today I get the wonderful opportunity to introduce to you two fantastic people that I've met over the years. One you might have seen at our mini summit at the University of California, Irvine, Kristen Kelly. She is a dermatologist, but she also does research, and that's important too. That'll play a key part in today's talk. But she's been a great friend. She's been one of our reunion of champions for dermatology, and we just love her to pieces. And then we have on the other side, no, it's not a boxing match, I promise. We have Doug Marchuk, who is a distinguished professor of molecular genetics and microbiology. It's a lot. Good job, Doug.
Oh,
And but he's one of our main researchers that always comes to the table when it comes time to plan our research meetings and work on different grants. I know they were just working on a Leduc grant and some other pieces of the puzzle. So with that in mind, welcome to both of you.
thank you.
Excellent. So I'm just going to get started and say, uh, Kristen, if it's okay, what got you into research and what excites you the most about what you do?
Well, I think the main reason I got into research, truthfully, when I finished my residency, so I finished some of my training to be a dermatologist, I went into mostly private practice, although I stayed doing a little bit of research at the university. And then after a couple of years, I went back to the university full time. And the reason I did that is because I would like to say that, every patient I see, I have the perfect treatment for and that I can cure them. But the truth is that I can't, and not that I don't want to or that I don't try, but we have limitations in medicine. There are some things we're able to treat very well, and there are some things that are more difficult to treat. And so the reason I like research and my main reason I came back to the university is probably I see more of the patients who have more challenging things to treat at the university, and then I have the opportunity to do research and try to find new ways that we can do a better job to treat people who have more difficult to treat disease. So I guess that's kind of why I came back to the university and why I do research. Now, research can be very slow, so it doesn't mean that like every day I come up with something I wish I could, but over my lifetime of working at the university, I hope to be able to contribute a little bit to helping some of the more difficult questions.
Awesome. Thank you. Doug, how about you?
Yeah, I've always been interested in science, and I think I wanted to be a fireman or a scientist or a rocket something, when I was a kid. But all joking aside, I have been interested in science for a long time, and I would say the reason why I got into research on human diseases, especially something that had a genetic type component, um, was because I think it really, that genetic window into the process allows us to understand what's going on really in a way that, stroke or something general, it's not possible. So very early on when I got my PhD in sort of hardcore molecular biology, and it had a very oblique, uh, you know, reference to disease and health. I really started to focus on disease beyond that and more medically relevant things.
So, that sounds exciting. So, Kristen, why Sturge-Weber? Why us? Oh, I think you're on mute.
Oh, I'm so sorry. I didn't even realize I hit that. My sincere apologies. So, I'm a dermatologist, so I treat skin disease, but more specifically, I'm a laser surgeon. So, I treat skin disease that we try to make better with lasers. And so, there are a variety of diseases that I treat with lasers, but the ones that I like to focus on, sometimes lasers are used for more cosmetic things, but I have really liked to focus on more medical conditions that can be that can get benefit from laser treatment. So severe burn scars, for example, vascular anomalies or birthmarks, et cetera, related to vascular changes. And so Sturge-Weber and then port-wine birthmarks in general have definitely fall into that category. And truthfully, I have worked at the Beckman Laser Institute for a long time, and that's been a strong center for decades, almost truthfully, I think it's now like 40 years, something like that. So we're getting on to half a century, not quite, but of, working with patients with port-wine birthmarks and Sturge-Weber syndrome. So.
Awesome. Thank you. How about you, Doug? Why Sturge-Weber?
Yeah, I started out working on a different truly inherited disease called hereditary hemorrhagic telangiectasia, and it also has vascular malformations, as Dr. Kelly really would certainly know. And the other name for that, by the way, is Osler-Weber-Rondeau disease, or if you're French, Rondeau-Osler-Weber, but we won't get into that.
C'est très bien.
I'm sorry?
I said it in French. That's
Oh,
great.
oui, très bien. So, the key is that Dr. Weber, I think, German, he's described a number of these vascular disorders. So I was very much intrigued when I was studying hereditary hemorrhagic telangiectasia. Could some of the things we learn have to do with Sturge-Weber? And so that's how I got involved.
Awesome. Doug, you had recently applied for a grant. What does that process look like to you?
Yeah.
And what would those steps look like?
Well, often when you're studying disease, one of the most important things is, and I am not a physician, I've never taken care of any patients, and I don't have any access to patients. I really to find strong collaborators and often clinical colleagues like Dr. Kelly. So, whoops. So one of the things I did, something happened. Okay.
I think
I'm back on, I think. Yeah. So, writing a grant, let's take, for example, did I go off? Yeah. Am I going on and off or not?
somebody was
Okay.
talking. I put them on mute, so we should.
Okay. Yeah. I just I just wasn't sure whether I was going in and out. I apologize. Anyways, to cut to the chase here, you would write a proposal if it was for the National Institutes of Health, which is the main funding body of the United States government when it comes to biomedical research, dealing with health and disease. That you may take a month or two of your life to write this grant, to get all the paperwork together, to get the science together, to get the preliminary data that would support that what you can do is feasible. And so it's a lot of work. There are also other agencies. Julia, you mentioned the Leduc agency, which is a international agency, and a group of us puts things together. We had conference calls and everything, and Zoom meetings just like this to come up with ideas, write it. Like an NIH grant, the science part is like 13 pages. But the grant itself could be a couple hundred pages because of all the budgetary issues and everything. But the core of it might be that science would be those 13 pages, but they're chock full of figures with data. I think that's good enough for now. Yeah.
I agree. It's a lot of work, is the summary.
Oh yeah.
And a lot of detail.
I wasn't exaggerating when I said that, you might devote a couple months of your life. I mean, the reality is that maybe in 1 month you knock it off, but you kind of, people often, scientists especially, they'll sequester themselves, they'll hide for a month to write this thing to get it picture perfect.
Yeah. Awesome. Thank you. Kristen, you also do research in the clinic, or in the clinical world. What is the difference between what you do and what Doug does?
Well, I think as he mentioned, actually, I think we try to work together, overall. And so, there's basic science. You can divide it into different categories. There's basic science work. So, kind of we call it bench research sometimes, so working in a lab, not necessarily with patients, although you might have a biopsy from a patient or something. So there are ways that you're working with patients, but the patient's not necessarily right in front of you, kind of thing. There's clinical research, which is kind of the other end of the spectrum, which is, trying something on a patient, or, a treatment, or a way of measuring things or something like that. And then there's translational research, which is really often what we're trying to do, which is kind of bridging that gap a little bit, taking what you find from the lab or the bench, as we call it, and then translating it or implementing it in the clinical side. So, I mean, I think a lot of what I do is translational research. I do a little bit of basic science with colleagues and then the clinical implementation of it. So I think really what we're trying to do is bridge that gap and bring it all together. So although there's some slight differences, it's really just kind of a circle of things that we're working together to accomplish.
Yes.
Absolutely. Absolutely. And what one of the reasons we wanted to do this today was, I know, Doug, when you had put in your grant, you had said that you needed tissue biopsy. And the first person I thought of right away was Kristen Kelly, because she always has, she thinks in that module. So collaboration is such a key word in our world, right? And so what does that mean to each of you?
I'll go first. Yeah, I mean, collaborations are just the key. I've had so many collaborations, not only with other scientists, but with other physicians and physician scientists like Dr. Kelly. So it's just the most important thing. I think the intersection of the bench to the clinic and the patient, you have to be in that inner space, and we have to work together.
And I mean, collaboration is just working together, right? And early in my career, I really benefited from some mentors who really taught me, you can try to do things all yourself, but honestly, then you're only looking at it from your small perspective and your small experience. And then, when you collaborate with people with different skills and different backgrounds and things like that, you get like a much wider perspective. We talk about diversity, and that's important in so many different aspects. But from a scientific standpoint, I know about skin, or at least about some things in the skin, but I don't always know about the newest molecular techniques or something like that. And I certainly am not a statistician, so we work with people who have expertise in all those different areas so that then we have experts in all those parts. And then whatever we do, we try to make it that much better because we have the perspectives of so many different people.
I have to say, having the meeting with the two of you to figure it out, and we have great news, right? We have an IRB that was just approved, but this week already. And it's very exciting because we've seen it full circle, so to speak. And it takes the patient, but it was very interesting to the rest of you on the call to hear just how everything worked, how much of the tissue, what does it look like, what are they going to store it in, and to have all the conversations around that was exciting to hear from a caregiver's perspective, and I'm sure as a patient's perspective as well, just to see how it all works, because we only know as a patient the other side. What do you need from us to make it work? So it was wonderful to see that part. So I share that with the rest of the group. One last question, I promise, because I promised them I wouldn't be too overbearing, but in a perfect world, based on your experience, if you had one question to answer or solve for, we prefer Sturge-Weber, but in your own world, what would that be? Doug, do you want to go first?
Okay. Yeah, no, if I had one question if I can rephrase it differently, I would love to see something that we've discovered on, in the laboratory, make it all the way to a treatment for patients, because it may be a really effective treatment. It may not be a cure, but it'll be a super effective treatment. And I'd love to see that happen. I study 3 diseases with that vascular component, and I'd love to see one of those. And if it's Sturge-Weber, I will be doing cartwheels in the hallway.
We need videos of that, please, for sure.
And we're probably both cheating with our answers a little bit, because I know you're probably looking for something more specific. But honestly, I mean, I think I would say, I was trying to think, and again, I think this is cheating a little bit. But what I would love to make the world a little better place for some people. You know what I mean? And so I know that that's not a very specific answer, but I mean, I think that there are a lot of things we can do. And for example, though, it's possible if we find a treatment that works for Sturge-Weber syndrome, that it might also help another group of people. You know what I mean? Or even if that particular treatment does, it might give us a clue about how another disease is different or something, and might help them as well. You know what I mean? So there are a lot of these things are pathways that are connected. And so, if we discover some information that is going to help people in the long run, I think that's what we both want to do. And, for the most part, I think that's what science should be and what most people want to do with it is just to try to help people or try to make the world in some way a little bit better. There have been some unfortunate episodes in history where perhaps it wasn't used that way, but for the most part, I think that's really what people are trying to do. And I think Dr. Marchuk and I are no exception here.
Awesome. Well, thank you both for that. Do you have any suggestions for us as patients walking in, to a study? What you would, I failed to see that question when I asked you the question. So what would be ideal for us as a patient to know walking into a study?
Well, I think, not every study is right for every person. You know what I mean? So, when you're getting ready to sign up for a trial, it is their obligation to go over a consent form with you, which should tell you about like what the study's for, why we're doing the research, what is going to happen in the study, and then any risks that are associated with the study. And so then each person just has to decide if that's right for them. And there's nothing wrong with getting the information about a study and then just deciding, you know what, this is not right for me. I don't feel comfortable with this and I don't want to do that. That's okay. But you can certainly ask as many questions you have about it. And, I will tell you as a researcher, maybe we don't know all the implications. We try to know as much as we can, but then it's also our obligation if something changes that we tell you that information, that we update our consent forms and then provide you that information. So I do think overall, sometimes people, not always, but sometimes people are suspicious of research. Like, are they trying to like test something on me? Do I feel like a lab study instead of just, like a human being. And I think overall, that's not what research should be. It's to, try to help people. But if you are feeling like that, I think you can let someone know. And I will tell you, like I say, for the most part, what studies are is to try to find something that we can help that is going to help more people. But, it has to be right for you as well.
Sure. Doug, do you have anything to add?
I would, if we do enroll some patients in some other studies for the hereditary hemorrhagic telangiectasia studies. And I would say if it's right for you, come with a sense of excitement and, maintain contact and say, how's it going? Realizing, of course, that it's not just, oh, we got your sample of tissue or blood or whatever, next week we'll have the answer and we'll have it cured. But come with a sense of excitement that we're working hard and people are working hard and are really dedicated. So come with that sense of excitement and come and partner with us in the same way that the physicians and the scientists and everybody in between is partnering. It's a partnership with the patients, and come with that sense of excitement and be part of the team, ask questions, and yeah.
Yes. And I know from both of you and others like you that the excitement is there. And that's always exciting with something so rare like us, and that it is wonderful when we can see your excitement. So thank you both. I promise no more from me questions, but I'm going to open the floor to the rest of you and see if you have any questions for either Kristen or Doug.
And it's okay if you guys don't have questions, but we just want to be available if you do.
For sure.
Okay, so it's Sabrina, and I don't know what the new grant is.
Oh, very good.
Okay, so there's a couple different things going on at once. And so let me tell you. So I wrote a NORD grant. There's the National Organization of Rare Diseases, and that was funded. It's quite small, unfortunately, and it's already started. And we realize, of course, that it's taken a while for the Institutional Review Board in California to approve all the paperwork and, going back and forth with questions. So we have those funds, and that grant, that NORD grant, is to really understand if there's anything else besides the original GNAQ somatic mutation going on in the tissue. And we have some ideas from our other studies, by the way, in the two other vascular diseases, that there's some chromosome mechanics going on that may be going on in the Sturge-Weber tissue. And so that's what we want to investigate, I should say. Yes. We're not testing anything. We're investigating that hypothesis.
Okay, thank
Does
you.
that help? Yeah. So that's why the affected tissue is so important. It was so important in the original discovery with Matt Shirley and Comey, and we were a major part of that as well, and doing a lot of validation sequencing and all sorts of additional samples. It was to be able to get that tissue because something's going on there, and that's
Mhm.
to go back and look. And yeah.
That's great. Thank you.
I have a question. What are some of the laser treatment options for people with vascular birthmarks? I know of the V-beam or the pulse dye laser, but what are some other options for people to use?
So the V-beam or that's a brand name of the pulse dye laser is one of my favorite ones to use, but there we actually are just getting ready to wrap up a study using a long pulse 532 nanometer laser, which I will tell you I don't have the final data, but I don't think is better than the pulse dye laser. I think it's equivalent, but is another option for people. Sometimes we have people who have, their birthmark has gotten thicker or they have nodules in it. And then sometimes we'll use like a long pulse 755 laser or a long pulse 1064, not my favorite to use, but for, specific nodules or something, sometimes we'll use that laser. So I think there are some others. What I think we're ultimately going to need is to have medications that we use in combination with the lasers. So a lot of the work that we're doing is to try to find, when I say we, I mean like the world of researchers, Dr. Marchuk and everyone included, where we're trying to figure out what the pathway is, we know it's a GNAQ gene change, but then what pathway is that affecting, what specific things are happening in that pathway so that we might be able to find medications that can stop those effects from happening. There are lots of examples in medicine. For example, probably the best one and the earliest one was tuberous sclerosis. People use medicine called rapamycin or sirolimus, that when you use that medicine, it can reverse a lot of the things that happen with the gene change there. So we don't have one yet for port-wine birthmarks or Sturge-Weber syndrome, but that's what we're trying to find is one that could work on the pathway that perhaps with a laser. So in patients with tuberous sclerosis, we still do laser treatments, but then we use the medication so that it doesn't come back as quickly and the effects are much more dramatic.
Okay, I have another question. Is there like I know that sunscreen's important, but how does sun and sunscreen affect port wine stain? And like, is there extra precaution, especially with sun care, that we can take?
Well, it doesn't necessarily specifically affect the birthmark. It's not like if you get more sun, the birthmark's going to grow more or anything. But when we're doing our laser treatments, the more pigment or melanin you have in the skin, because it's on the surface of the skin, it kind of blocks the laser from getting to the blood vessels in the port wine birthmark. So that's why when people are having treatment, we ask them to do good sun protection because we want the laser light to be able to get to the blood vessels and not be trapped on the surface of the skin from the pigment. So that's why we do that. And of course, it's the same sun protection we would say for someone who gets sun damage. I live in California, so we have basically everyone has some sun damage kind of thing, right? And so, it's the same ones that we use. We know that physical blockers that have, for example, iron oxide and things like that can be even better than some of our other sunscreens. But, just trying to protect the skin and make it safer and more effective when we do our laser treatments. But it doesn't hurt the birthmark itself, the sun.
Anybody else?
Yeah, this is Jamie. I have a question. Is there any long-term studies going on about developing complications with port wine stains? I had a spontaneous brain bleed. They didn't connect it to that, but I've had other stuff with pregnancies too that I think, sorry, that's my son screaming with a video game. I'm so sorry.
Okay.
That I think it was related to my birthmark, to my port wine stain.
Do you want to answer that, Julia? I mean, I know that, Julia, I'm sorry, I shouldn't answer for you, but I mean, right, I know that some of the groups have talked about looking at, monitoring long-term changes, both skin standpoint and then neurologic and eye standpoint, or Dr. Marchuk, I'm sorry. I should just be quiet and let you guys answer.
So we have something called the brain. Well, I'm part of a larger, very large National Institutes of Health grant called the Brain Vascular Malformations Consortium. And Jeff Loeb, who's a neurologist, sees a lot of adult Sturge-Weber patients, and he's really interested in those very questions, not so much, one particular patient's outcome, but what the downstream analysis, not so much the port wine stain on the face is influencing the brain, but there are changes also in the brain vasculature, and those lead to changes in the brain parenchyma. So Dr. Loeb and Dr. Juhas at I'm blanking. Henry Ford Hospital, I
Yep.
think. I'm forgetting, but somewhere in Detroit, they're working on some of those monitoring longer term effects, basically following patients for their brain involvement. So it's not that the wine stain itself has an effect, but the fact that there's concomitant changes in the brain.
Mhm.
Yeah. And so people are looking at that kind of thing. And Dr. Loeb has some ideas about calcium, and we won't get
Yep.
into all that, but yeah. So yes, people are thinking about that.
They
I
are.
mean, I'm sorry, what would be your recommendation? Because that happened 2 years ago. So of course I'm always worried it's going to come back again. Is there a direction I should go? Like you said, maybe that Dr. Lowe or somebody that can look into it? Are there certain kind of scans? Obviously, I get CAT scans and MRIs and stuff, but anything I can look at to see if there is a correlation between the port wine stain and future issues that could occur?
So Jamie, I'll take that for you. Yeah. Where are you located?
I'm in Atlanta, and I think I've called a couple of times, but
Sure.
Emory is the closest thing, and I just don't feel they're as experienced as other people out there. I mean, even these two wonderful doctors on here now, I just don't think they know enough about it.
Okay.
Because I went to them with my other issues when I was pregnant, and they just didn't really want to listen to me that it was my birthmark, and I knew it had to do with my birthmark. So,
Yeah.
mhm.
Well,
That's
Dr.
hard.
Jeffrey Loeb is in Chicago,
Mhm.
and
Yay.
Dr. Juhas is in Detroit somewhere, one of those
Yep.
hospitals.
At Wayne.
And I'm sorry, yeah,
Oh, he's at Wayne. Yeah. Go ahead.
yeah, and Wayne State.
Yep. Wayne
That's
State.
not Henry Ford Hospital. Okay, any event. Those two guys, Loeb, I don't know. I hesitate I would never give medical advice, thankfully, but I do have tremendous respect for the people who I've interacted with who are doing some of this clinical research. And Dr. Loeb and Dr. Juhas. Dr. Juhas has shown some beautiful case series. He's even published on these. But in our Brain Vascular Malformations Consortium, notice it's about the brain vascular malformation, which is also there. It doesn't mean that it's because of the face.
Yeah.
He's done some beautiful things about the changes that occur over time. And so he's really studying, and that's where I would think to look. And
How
Dr.
do
Loeb is really he's really kind of treating, I mean, he sees it as like, and rightfully so. I've forgotten where we were. I think it was not in Charlotte, but the Sturge-Weber group was in, I believe it was in Dover, Delaware, or whatever. We
Good.
had a big meeting, and Dr. Loeb was asking, well, what do you adult patients want to talk about? And then
Mhm.
they came on with that, and that was really telling, and it's influenced our NIH grant, which unfortunately is ending
Yeah.
in next summer or spring even.
So,
So I work with I'm a patient of Dr. Loeb's for
Excellent.
the past, I don't know, 7, 8 years. And I love what he's trying to do, and I help as much as I can, but I would highly recommend um, um, contacting Gina. That's his assistant. She's fantastic, and she can help you get information.
Jamie, can you email me and I'll get you in touch with them tomorrow?
perfect. Thank you guys so much. I appreciate it.
One thing I will say is I really appreciated what you said, Jamie, because in all of the work that we've done with all these vascular malformations, you see these people who have hit this, like they have this long odyssey of trying to find the right person, and once they hit on the right person, and so there's some really dedicated people. It's just got to find them, and that's where the foundation really knows what's going on. That's why I like Julie. Yes, Where are you? Oh, I'm in Atlanta. Well, let me see what I can find there. Yeah.
Well, it is funny, Doug. I was just thinking of Jack that we were talking with last week.
Jack.
Then yeah, Jack Arbisser, who was we were just talking with last week about something different. And I was like, oh, I could call Jack, or let's get you involved in the Brain Vascular Malformation Consortium, because there's still openings to get in there. And wink wink, sometimes US will sometimes look at MRIs if say we maybe ship them to him and give us, his opinion too, because he's our guru of MRIs. So he's lovely when it comes down to it.
And Dr. Loeb is also I don't know what the right word is, the head of the medical
Clinician.
group, the advisory board or whatever.
Yep.
Yeah. Yeah.
We'll get you squared away. The nuts and bolts. So we heard from some of you. Does anybody else have a question? I don't know if
Yes, this is Lori. I have a question actually for Dr. Kelly. I have an adult son with a very extensive port-wine birthmarks, and he gets treated. He's been treated by several different dermatologists just because his dad was in the military, so we moved around a lot. So now he's an adult doing it on his own. His current dermatologist doesn't really cover his eyes during treatments. And I was wondering what your opinion is. He has glaucoma, so as a parent, I'm very concerned about his eyes. How do you feel about lasers and eye protection?
I'm a little bit confused because whenever you're doing a laser treatment, you have to do eye protection. So what kind of treatments are they doing?
They're using the pulsed dye laser.
And they're using no eye protection?
They put a cotton ball on his eye.
Well, I would say that kind of standard of care would be to do appropriate eye protection, and that would be either laser safe, depending on where you're doing. If you're going to treat the eyelid, then you have to put a corneal shield in. It's kind of like a little metal contact lens.
Right.
If you are going to be not treating the eyelid, but on the face, there are either laser safe iPads that you can use, or like metal goggles. They look like kind of like swimming goggles, but they're metal so that they're making sure that they protect the whole eye. But I do think, and then if you're treating totally off the face, then you can use certain goggles like the laser surgeon uses. But I cannot emphasize enough that the appropriate eye protection is really important.
Okay, and will you go pretty close to the eye with, say, the goggles or the iPads? You feel
No.
No. Okay.
I can go, well, let me say, I can not with the goggles, I only use the goggles if I'm treating off the face. If I'm treating on the face, then I might use the laser safe iPads, or like I say, the swimming kind of goggles that go just right over your eyelids. I wish I had a picture of them to show you, but that go right there. And then you can go pretty close to the eyelids. But you just have to go, you can't go closer than what is covered, of course. But if you're going to be treating the eyelids, then you need to put the corneal shield on. But that's not a problem to do. It's relatively easy. I did it like 3 times today. I put a couple little drops of numbing medicine in the eye. I put a little lubricant on the shield, and I can get it in like literally 2 seconds.
Okay. All right. Thank you.
Good question. Anybody else?
I have a third question. If anybody else has one, please ask, but I'm just curious. What kinds of research, I know of one in Boston, but what kinds of laser research developments are out there that we as Sturge-Weber patients or port-wine patients can take part in?
Well, there's lots of trials. I mean, I just was mentioning we had the kind of comparing the pulse dye laser to the long pulse 532 nanometer laser. We're almost done with that trial. We're just doing a couple follow-ups. I think, there are hopefully soon going to be some new medications that we're going to be trying in combination with laser. We've also done a number of trials. Actually, one just came out in the Journal of the American Academy of Dermatology just last 2 weeks ago, I think, where we looked at patients who'd had laser and then how that changed the size of the blood vessels and things like that. And then actually there's going to be another study coming out with the University of Wisconsin looking at that and things. So they're all different kinds of trials. Now, do I think they're right now? And so then there's another one, sorry, which is photodynamic therapy. That is a trial. This is something that's done in China, but really has not been done in the United States very much. I had an NIH grant a number of years ago where we did a couple of patients, but this is where they give you a medicine, an IV medicine, so right into your veins, where we give a medicine, and then when we shine a light in a certain part of the birthmark and that medicine's there, you can get an effect. The nice thing about that is I mentioned earlier that if you have more pigment in your skin, it can be hard for the lasers to work because the laser light can't get down to the skin. This kind of treatment does not depend on the amount of melanin in the skin, so it can be done on patients with all skin types. However, the significant downside is that medicine that they give you into your arm sticks in your system for a couple weeks, and you cannot go outside. You have to literally stay inside for a couple weeks. So I think in an era of Zoom, that's possible for some people, but still difficult to do. But there is a multi-center trial that number of people across the country are doing for photodynamic therapy. So other than that, to my knowledge, I don't know of any like totally new devices people are looking at right now for port-wine birthmarks. But again, I think we're all very hopeful that we're going to find a medication and that we would definitely do a trial once we have a really good candidate.
Okay, I have another question. And this might be neurological in itself, but about hormones and how hormones have affect Sturge-Weber, because that's when all of my issues with Sturge-Weber started, is when I went into menopause. And I'm wondering how that might affect the skin. Other than, I mean, I know you get like your skin gets drier and stuff like that, but I'm wondering if it has anything else to do with it.
I'm not sure I totally asked the question, but you're saying that whether some of the other symptoms can affect the skin. Is that what you're asking?
Yes.
Yeah. Well, I think it all kind of goes together. I don't think skin dryness is necessarily a result of, having a port wine birthmark or Sturge-Weber syndrome. But I will say that, for example, in the port wine birthmarks, there's a well-known phenomenon where people can get like eczema in their port wine birthmark, which is kind of like dryness. And why that happens, we don't really know. But it probably is that the skin's a little bit different there. And for some reason, sometimes eczema will be only there and not on other parts of the skin. So
that's great, because actually I'm having that right now.
Yeah. And so that is a well-known phenomenon. We have a name for it, but it doesn't really matter. The point is just we know it happens. Just like also sometimes people get bleeding nodules, which is, kind of extra blood vessels that are forming there. And that can happen only in the port wine birthmark skin. So it is true that the skin can be a little bit different or have some of these other conditions. And I wish I could tell you I knew why that happens all the time, but unfortunately we don't know all of that yet.
Well, this was helpful. Thank you.
But Doug mentioned earlier about the Brain Vascular Malformation Consortium, that very long thing that we've been working on. So please, 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 if you haven't already signed up, please contact me in the next 24, when you can. I know the holiday's on Thursday, so I don't want to say in 24 hours, but soon, just so that we can get you into the study, because these pieces that we talked about today are critical too, because we don't know who else is experiencing it. And we're going to be able to, there's some really fun conversations we had earlier, Doug, you were part of too, that, they're starting to analyze the data, and it's really fun to see them get so excited and then ask, what do they want to study next? And, or what question are we trying to answer with these details? So this is really a key time to be a part of something really great, to take on new questions. So please, 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 if you haven't gone, and it's not painful, I promise. Marissa and I did it, and you just sign some papers, you get some files, and you send it to them, and they do all the work from there. Thank goodness. So it's one time we don't do it all, so we're good. So any other questions tonight? No, we're good. Well, thank you both. I'm not going to keep you. You got 15 minutes back of your night and go have dinner and good times. But Doug, Kristen, thank you for joining us this evening. And thank you for your question time and all your beautiful answers. And to the rest of you, let's have a great Thanksgiving, and we'll see you again soon.
Okay, thank you for getting this together. Bye.
Okay.
Thanksgiving, everyone. Bye-bye.
Thank you.
Thanksgiving.
Happy Thanksgiving.